The title of my project is Massteria, determining the wearing time and bacterial contamination of face masks with and without toothbrushing. I chose the title Massteria because I wanted to incorporate the two concepts of mask wearing and bacteria growth in a single word to capture people's attention. I chose this project because during the COVID-19 pandemic, masks have evolved as an essential part of our daily lives. A recent study done in the UK has shown that over 60% of people reuse their reusable face masks without washing them, and only 13% of people wash them regularly. This study has also found that 56% of people wear disposable face masks more than once, and 7% of people never throw them away. There are no clear guidelines on how often we should change or replace our masks, and given that we have to wear masks every day, I thought that this topic would be relevant to our daily lives. The purpose of this experiment was to determine how much bacteria accumulates on a face mask after a certain amount of wearing time, and to determine if brushing your teeth prior to wearing a face mask decreases the amount of bacterial contamination on the inside layer of the face mask. First, I prepared the agar. To make 10 agar plates, I placed 8 grams of agar powder in a clean glass measuring cup and added 375 milliliters of distilled water. Then I stirred the mixture until the agar dissolved completely. Then I brought it to a frothy boil on the stove. Then I let the mixture cool, and then I poured the mixture into the petri dishes until it covered the full bottom of each plate. Now I made the incubator. I got a styrofoam cooler, a basic lamp kit, and a 25-volt light bulb. And I made the incubator described in Science News for students. For the first experiment, I labeled each petri dish. On half of the dish, it was labeled inside, and on the other half, it was labeled outside. All 4 of my participants wore face masks for 2 hours and 4 hours and completed 3 trials. 2 of my participants wore blue disposable masks and the other 2 wore reusable cloth masks. Then I swabbed the inside and outside layers of each mask and plated them on the agar. I counted and recorded the number of bacteria colonies, also known as CFUs, colony-forming units, on each petri dish every 8 hours for 48 hours. For the second experiment, I labeled each petri dish. On half of the dish, it was labeled toothbrush, and on the other half, it was labeled no toothbrush. All 4 of my participants wore face masks for 2 hours and completed 3 trials, with and without toothbrushing. Then I swabbed the inside layer of each mask and plated them on the agar. I counted the number of bacteria colonies on each petri dish every 8 hours for 48 hours. For both experiments, I took pictures of all the plates at each time interval and recorded the bacteria characteristics like shape, size, texture, form, and color. In the 2-hour wearing experiment, it was found that the average bacteria growth on the inner layer of the 2-hour mask was 1.6 CFUs per centimeter squared, compared to 3.2 CFUs per centimeter squared on the 4-hour mask. The average bacteria growth on the outside layer of the 2-hour mask was 0.9 CFUs per centimeter squared, compared to 3.3 CFUs per centimeter squared on the 4-hour mask. For both layers, there was at least double the amount of bacteria growth on the 4-hour mask. However, I discovered that on the inner layer of the 2-hour 4-hour masks, there was more bacteria than what I was expecting. As seen in the 2-hour wearing experiment, there was more bacteria on the inner layer compared to the outer layers at 48 hours. Since I saw this, I decided to do more research and do an experiment to see if we could cut down on bacteria growth on the inner layer of our face masks, because I found that some participants were complaining about skin issues due to wearing a face mask, and I thought this could be related. I did this by seeing if brushing your teeth prior to wearing a face mask would make a difference in bacteria growth than not brushing your teeth, and it does. For the second experiment, with and without toothbrushing prior to wearing a face mask, the average bacteria growth on the toothbrush masks were 1.1 CFUs per centimeter squared, compared to 2.4 CFUs per centimeter squared on the non-toothbrush masks. I saw that when participants brushed their teeth prior to wearing a face mask, it resulted in half the amount of bacteria growth on the inner layer of the face mask. The average bacteria growth at 48 hours for each participant's masks, with and without toothbrushing, was compared. For 3 of the 4 participants, the average bacteria growth was at least 50% less on the toothbrushing masks compared to their non-toothbrushing masks. For trial 1, participant 4 had significantly more bacteria growth on his with and without toothbrushing masks. For his first trial, participant 4 had 3.5 CFUs per centimeter squared on his toothbrushing masks, and the average for all the participants that trial was 1.5 CFUs per centimeter squared. For his non-toothbrushing masks, participant 4 had 4.4 CFUs per centimeter squared compared to the average of 2.1 CFUs per centimeter squared. It was identified that participant 4 had a beard for the first trial, and following shaving his beard, his CFU counts were similar to the average of all participants in trial 2 and 3. Overall, it was found that when participants brushed their teeth prior to wearing a face mask, it resulted in less bacterial growth on the inner layer of the masks. Based on the findings of my experiment, my results can be applied to the real world by making some recommendations on how we should wear and care for our face masks. Some recommendations are: change masks every 2 hours, brush your teeth before wearing a mask, discard disposable masks after each wear, wash your cloth mask and dry completely after each wear, and wash and sanitize your hands before touching your face mask. These recommendations can help avoid the possibility of contaminating the environment or yourself with bacteria. More education will be required to educate the public on the issues of face masks and bacterial contamination, and the importance of changing and washing your face mask on a regular basis, the importance of oral health and hand hygiene. The most difficult part of this project was counting the CFUs manually, since some of the agar plates had more than 200 colonies, and I could not tell if the CFU was an individual colony or multiple colonies. One way I solved this issue was counting and taking pictures of all the agar plates every 8 hours instead of 24 hours, so it was easier for me to see and compare the CFUs over time. Also, if the agar plates had more than 100 CFUs, I had an additional person count the CFUs to confirm my number. Future development could be done to see if there are any different ways to cut down on bacteria growth and contamination of face masks. This could include studying the different types of toothpaste, mouthwash, or face products used prior to wearing a face mask that could decrease the amount of bacteria growth. Also, future development could include developing an antimicrobial face mask, which reduces the amount of bacteria growth on the mask itself. Also, in this experiment, it was found that previously washed controlled masks had more bacteria than expected. So it would be important to research the best washing methods for reusable face masks. It would also be important to study if reusable face masks are still effective after a certain amount of wearing time and wash cycles to determine when a cloth mask should be discarded. I used a scientific method throughout my project, specifically when I was writing my procedure. Since I was writing my procedure myself, and there was very limited research done on face masks and bacteria growth, I had to do a couple pre-experiments before creating my final procedure. For example, the first time I wore my 2-hour forearm masks and then swabbed it, I swabbed the petri dish about 3 to 5 times and then checked the bacteria growth after 24 hours. And I could not count the colonies since they were not distinct. So I researched how to swab petri dishes and then decided to check my petri dishes every 8 hours instead of 24 hours. I also used a scientific method to develop my second experiment, determining if brushing your teeth prior to wearing a face mask could cut down on bacteria growth on the inner layer of the mask, based on the results from my first experiment. In conclusion, these experiments show that the bacterial counts doubled with wearing time and that brushing your teeth prior to wearing a face mask decreased the amount of bacteria growth by at least 50% on the inner layer of the face masks. Given these findings, it is recommended to change your face mask every 2 hours, brush your teeth before wearing a face mask, and the importance of good hand hygiene following wearing a mask to avoid contamination by bacteria.